Hi everyone, my name is Erin Newcomer. Thank you for having me today. Today I'll be discussing about how standardized hygiene interventions are needed to reduce sink drain antimicrobial resistant organism reservoirs. And so we'll start out with just a little bit of background. Antimicrobial resistant organisms, or AROs, can live on different hospital surfaces. And when they live on these surfaces or environmental reservoirs, they can transmit to patients and cause healthcare associated infections in those patients. And this is something that has been frequently reported on in literature, particularly related to reservoirs surrounding sink drains or hand washing sinks or water sources in hospital systems. And so my goal today is to talk through two different projects that I've been involved in that are investigating these bacterial reservoirs in hospital systems. And so the first project is kind of a who's there and what are they doing project. We're looking at what surfaces can serve as bacterial reservoirs, what sort of spatial and temporal dynamics do these organisms have, and then which ones are the ones causing patient infections. And then later on, we'll get into project two, where we look more into how can we solve this problem? How can we remove these bugs? How effective can an intervention be, and then what happens after an intervention is stopped. So we'll start out with just project one, looking at who's there and what are they doing. So in project one, we collected surface and patient samples over time. We had the opportunity to sample an old ICU for its last month of operation before it was closed down, and then also a new ICU before patients and staff moved into the ICU, and then across its whole first year of operation. And so at each of these sampling time points, we collected swabs from several different ICU surfaces like sink drains, sink surfaces, the floor, bed rails. And then in total across this study, we collected nearly 1,600 samples. And for all of these samples, we selectively cultured these 4 different antimicrobial resistant organisms. And then once we identified these isolates, we then subjected them to whole genome sequencing. And so first, I want to just talk about where we are finding these AROs on these surfaces. And so what we're going to look at here in just a second is how many isolates we were able to collect from a given sample at each of these locations along the y-axis here. And so when we look at this data, what we can pretty quickly see, the dark gray are these isolates, by the way, and the light gray is if a sample was not collected at that site. Is that the vast majority of the isolates we collected in this study are coming from either the in-room sink drains or the bathroom sink drains. We do find a number of other isolates from other locations like the floor, the nursing call button, and different locations in the room, but the vast majority and significantly more isolates were collected from sink drain sources. And another difference we found was that there were significantly more isolates found in the sink drains in the old ICU before it shut down than were found in the new ICU sink drains across that first year of sampling. Something interesting I do want to point out, though, is that we actually did find a number of isolates in the sink drains in this pink area, which is the before patient and staff move in. And we actually didn't find a significant difference before patient and staff move in compared to the new ICU after patient and staff move in. And so next we wanted to look at what these organisms are. And so what we're looking at here is the most frequent genera of isolates collected from the different sites we collected over the course of this study. And so you can see the vast majority of our isolates were either Pseudomonas or Stenotrophomonas isolates. And these isolates are something that we've seen a lot in literature in this context, actually. Because these two genera can form what we call biofilms in wet environments, which are kind of just more resilient groupings of these species that can survive in wet environments like sink drains, but they can also resist antibiotics and cause patient infections. And I think I might have forgotten to mention this, but also across the course of this study, we collected clinical samples as they were available as part of routine care. So we also have isolates from patient stool and bloodstream infections. And so for the purposes of this presentation, I want to just focus on those Pseudomonas isolates we collected. So looking even more detailed, we found that the majority of these isolates were Pseudomonas aeruginosa, which is a common healthcare-associated infection. And so kind of one of the first things we like to look at genomically at these is a phylogenetic tree. And so if you're not familiar with phylogenetic trees, You can kind of think of this as like a large family tree where every tree branch tip here is an individual isolate. And then isolates that have shorter branch distances between them are more closely related, and isolates that have longer branch distances between them are less closely related. And we can also visualize different metadata around the phylogenetic tree. So we have different colors associated with different metadata on these kind of circles of colors surrounding the tree. And something that we found very interesting when we look at these samples phylogenetically is that we found several clusters of Pseudomonas aeruginosa that seem to be related between the sink drain isolates collected and isolates that we collected from patient infections. So I'm going to zoom in on just this small portion of the tree down here, which is this MLST 1894. And you can see that we have a lot of very closely related isolates in here, many of which are from sink drains, which are these dark blue and light blue rectangles. But we also have several isolates in this cluster that are these red rectangles, which represents isolates collected from patient bloodstream infections. And so this is something that we're very interested in since the goal here is to reduce patient infections. So this was kind of our first thing we wanted to investigate more closely. And so to do this, we wanted to define strain groups because just because something is the same species, we don't necessarily know that they are what we consider the same lineage or strain. And so we first just looked at the pairwise SNP distances between all of our Pseudomonas aeruginosa isolates. And so that's what we're looking at here is these SNP distances and then the number of comparisons we have for these isolates. And so you can see we kind of get this distribution where we have a larger kind of hump of comparisons in the middle here, and then two different groups, either at very low SNP distances or very high SNP distances. And so to just kind of get a broad look at similar strains, we set a pretty loose SNP cutoff of 2,743 SNPs. And for those of you in the field, you probably know this already, but 2,700 SNPs does not indicate that these are necessarily the same strain, just that they are more closely related than other comparisons within this species. And so with this kind of loose SNP cutoff, we were able to define a number of strain groups of Pseudomonas aeruginosa. But one thing I would like to point out is this strain group 1, which is our largest strain group. All of the isolates within this strain group were actually within 11 SNPs of one another, so much more closely related than that 2,700 SNP cutoff initially. And so that's the strain group that I'd like to focus on for this analysis. So we wanted to look at longitudinal strain tracking of these isolates. And so what I'm showing here is a little bit of a complicated figure, but it is basically looking at appearances of these different strain groups over time and at different sites that we sampled. So I really want to focus on strain group 1, which was that large strain group that's all within 11 SNPs of one another. And these are represented by these yellow squares. And so you can see these yellow squares show up actually before patient and staff move in. So in this pink area right here, it showed up in 2 different patient room sink drains and then proceeded to remain in those patient room sink drains over the full course of the year of sampling we conducted here, and then also appeared to spread or at least show up in all of the other room sink drains as the year progressed. And so this is really interesting because a lot of kind of our theorizing is that these organisms are initially seeded from patients. And so it's really interesting that this showed up before patient and staff move in. In addition to this, I actually would like to also point out down here we have where these different strain groups may have shown up in patient clinical specimens. And we actually found this group 1 of Pseudomonas aeruginosa showed up in 3 separate patient bloodstream infections. And so these occurred after this lineage was seen in the sink drains. So this gives us, it is plausible that these isolates could have transmitted from the sink drains to the patients themselves. And so our main takeaways from this project are really that these opportunistic pathogens like Pseudomonas aeruginosa can form long-term reservoirs in sink drains, as we saw it colonized over the course of a year, and they can also cause patient infections, as we observed. And importantly, this is happening in non-outbreak settings. Many different outbreaks have been published on of transmissions and contamination happening from sink drains in outbreak settings. But in this case, this was not a known outbreak setting. So what we are observing is that this could be happening in more places than we might currently realize. And so because of this, we need to develop protocols to reduce antibiotic-resistant organism burden in hospital sink drains because it can be causing these patient infections. Moving right along into project 2, we now can address this question of what can we do about these bugs, these AROs that are living in sink drains, and then evaluating a potential intervention for its effectiveness, and then also what happens after the intervention stops, because that can provide us with some additional insights as well. So there have been a variety of different interventions that have been proposed and tested in literature, but there are pros and cons that come with every intervention. And so some of the ones that have been previously used are water filters added to sink faucets. However, this does miss any previously existing downstream or sink drain colonization, which from what we observed seems to be a significant factor in this transmission. It has also been proposed to go completely waterless and remove sinks and sink drains from patient rooms altogether. However, this can have impacts on patient care and can also be very expensive for previously existing hospitals and room setups. There have also been a variety of chemical interventions that have been proposed, such as using acetic acid or bleach and pouring these into sink drains to combat these organisms. However, these have shown in some cases limited effectiveness, and this is largely because of the ability of these organisms to form biofilms, as we discussed before. These different chemical interventions aren't necessarily able to break down these biofilms in the time that they are in contact with them. So, in an attempt to resolve some of these issues, we propose to use a different chemical intervention that involves a foamed peracid-based disinfectant. And so I want to look at a couple different figures that have been published by other groups that are kind of why we decided to use this approach. First, we're just looking at a graph that's looking at recovery of bacterial growth after treatment with either hydrogen peroxide, bleach, or control that was not treated at all. And so the bleach is this the bleach is the dashed line in the middle here, which shows there was definitely a decrease in the amount of bacterial growth in the sink drain immediately after an intervention, but then increased back up to its original starting point. Meanwhile, the hydrogen peroxide, which was the solid line, seemed to more effectively reduce the burden of bacteria in this sink drain and have a slower return to the original level of growth. And so we're first looking and seeing, okay, maybe hydrogen peroxide can outperform bleach. Another group also looked at the use of hydrogen peroxide or peracid-based disinfectant and to look at the difference between just pouring the disinfectant down the drain and letting it wash past, or foaming the disinfectant, which could allow it to increase contact time and possibly help break down those biofilms. And so in this study, they did observe that foaming the disinfectant seemed to slow the return of the bacterial growth and provide more days of decreased bacterial growth in a sink drain. So this was our intervention of choice. And we implemented 2 different frequencies of this intervention on hospital sink drains to observe the effects on bacterial growth. And so, as I'm showing here, we had a set of control rooms that received no extra intervention. We also had a 1 time per week group that just received this foamed peracid intervention once per week. And then we also had a 5 times per week intervention. And at each intervention time, we pumped the peracid-based foam into the sink drain and allowed it to have a contact time of 3 minutes before flushing it out. And then we also wiped down the sink surfaces with a 10% bleach solution. And so in this study, we specifically wanted to investigate the impacts of this intervention on gram-negative bacterial burden of sink drains. And this is because gram-negative bacteria have been kind of the main culprits when it comes to these sink drain reservoirs and causing patient infections and outbreaks. And then we also specifically are going to look at Pseudomonas aeruginosa burden in sink drains. And then finally, we wanted to look at the return of these ARO interventions to see if it could help give us some insight into why these reservoirs are happening in the first place. And so we conducted this study in 18 rooms. And so we had 6 control rooms, 6 1 time per week intervention rooms, and 6 5 time per week intervention rooms. And at each sampling time point, we collected a set of different environmental samples. We collected swabs from the raised counter, the surrounding counter, the faucet, the sink bowl, and the sink drain. And we also frequently collected water samples throughout the study to ensure that none of this colonization was coming as a result of water contamination. And so we conducted this study across 5 separate phases. We started with phase 1, where no intervention was enacted, and this was to just get like a baseline level of what the colonization looks like at these surfaces. And then we went through two rounds of a phase of implementing the intervention for several months, and then stopping the implementation of the intervention to see what happens after the intervention is stopped. And across this, we collected a total of 31 time point samples. So first, just looking at kind of general results, similar to the last study, we were able to see that we collected bacterial growth from a number of different surfaces. So what we're looking at here is just the fraction of room sites where we recovered any bacterial growth in a given week. So across all of these sites, we saw growth from basically all of these sites frequently. However, when we conducted selective culture, we found that the vast majority of the antibiotic-resistant organisms came from sink drains, so either the inpatient room sink drains, or we collected samples from the housekeeping closet and soiled utility closet sink drain as well, and saw the majority of our AROs coming from those sites. And similar to the last study, the Pseudomonas aeruginosa and Stenotrophomonas multifila once again dominated these sink samples. You can see the vast majority of isolates we collected were these organisms. And so the first thing we wanted to look at was just if this intervention is reducing the number of sink drains that are yielding gram-negative growth. And so we first have our control rooms across these different study phases. And you can see there's some variability on the fraction of rooms that are yielding growth, but no significant differences across these different study periods. So next, looking into the 1 time per week intervention weeks, we see possibly starting to see some decreases in those phases 2 and 4, which is where the intervention was implemented. However, nothing quite reached statistical significance. And then finally, looking at our high frequency 5 time per week intervention, we saw significant decreases in the fraction of sink drains yielding gram-negative growth in those intervention periods and also in those follow-up non-intervention periods. So we're potentially seeing some longer lasting effects of this intervention over time. Next, we wanted to look specifically at Pseudomonas aeruginosa, since this is that most common isolate that we're seeing come from these sink drains. And so we just wanted to look at each sampling time point, the fraction of sink drains that we were able to recover Pseudomonas aeruginosa from. And so over the course of the study in the control group, we saw a variable number of sink drains yielding Pseudomonas aeruginosa, but pretty consistently getting it across all phases of this study. However, when we start to look at that 1 time per week intervention, we can see that in those intervention periods, we see several weeks where no Pseudomonas aeruginosa is recovered, which is really exciting. Even though none of these quite reach statistical significance, still very promising at this 1 time per week intervention. And then moving right along into the high frequency intervention, we can see nearly no Pseudomonas aeruginosa was collected from sink drains during these high frequency intervention phases. And following out of those intervention phases, we saw several weeks where still no Pseudomonas aeruginosa was recovered, which is really exciting because, again, we might be seeing more longer lasting effects of this intervention. So at this point, we have just a few remaining unanswered questions. We've seen that these AROs can be reduced by the intervention, but what we don't know is how long those specific AROs have been maintaining their reservoirs and potentially coming back after the intervention. And then those long-term effects. So at this point, we just have a few more remaining unanswered questions about the dynamics of these AROs, because we know they can survive long-term in these sink drains. However, we want to know how long are they maintaining these reservoirs? And then, like we just observed, after the intervention, we do see these ARO species returning, but we really want to know, is it those same strains of bugs that are coming back, or are these potentially new ones colonizing after the intervention is stopped? And so again, we decided to use whole genome sequencing to answer this question. So here we're looking at another phylogenetic tree, and this is actually containing the samples from both the Project 1 study and the Project 2 study. because the Project 1 study was only in 6 rooms, but those 6 rooms were actually also used in the 18 rooms in the Project 2 study. So we have some very longitudinal data here to look at. And so in this phylogenetic tree, we see that these samples largely cluster by MLST, which is totally expected in a phylogenetic tree. However, we also see that there's a lot of mixed clustering between the two studies. There's not really a divide between the two studies. And in particular, I want to just look at this top right corner here. These are largely that MLST 1894 that we've seen a couple of times here. And you can see a lot of these sink drain samples are coming from both the previous first study and this new study that we're looking at. So we wanted to look a little more closely into sort of these patterns. So once again, we decided to use SNP distances and a SNP cutoff to determine lineages. However, in this study, we wanted to be a bit more conservative with our approach. So we looked at those core genome SNP comparisons and set a very conservative cutoff of 7 core genome SNPs to determine if 2 samples are in the same lineage or not. And that is well within sort of the expected evolutionary rate of Pseudomonas aeruginosa in this context. So once again, this is another temporal strain tracking figure. I'll kind of walk through, there's a lot of pieces to this. So we're actually looking at strain tracking across that Project 1 previous study over here on the left, and then this current study here on the right, and you can see the 5 phases of this current study. And so we're looking at these different strain groups and how they appeared in the original 6 rooms of study 1, and then also in all of the rooms in study 2. And the one I particularly want to pull out is these green dots, this strain group 63. This is that MLST 1894 that we've seen so much of. And particularly, I just want to show that you can see it showing up very early in the previous study. As we discussed, this strain group actually showed up prior to patient and staff move-in. And you can see it occurring all across the previous study and then also occurring throughout the course of this current study in the control room and even in the 1 time per week rooms. And so across both of these studies is actually about 4 years of colonization. And of course, we had a little bit of a gap between the 2 studies, and we're not sampling all the time, but it seems very likely that this colonization is lasting for several years at a time. But once again, I just want to point out, since we did do interventions in this study, you can see in the 1 time per week and particularly in the 5 times per week interventions, these strain groups that are showing up regularly start to show up less frequently during those intervention phases. And in particular, I think it's interesting that in the control rooms, you see the same strain groups throughout the course of the study, no strain replacement really being observed. But in these intervention rooms, you can see different strains showing up at times after the intervention has been stopped, potentially replacing the strains that were once in those sink drains. And so we wanted to just quantify these results. And so here we wanted to look at the proportion of Pseudomonas aeruginosa strains and isolates that we're collecting, if they are new strains to a sink drain or if they are recurring strains. So first, to just kind of situate this data, we'll look at just the control rooms. So obviously in phase 1, there was no phase prior to phase 1, so all of the strains we just called new. But as we look through the other phases, it looks like largely we are seeing similar strains showing up in the sink drains that we initially saw. The vast majority of these strains are recurring. They were previously seen in a sink drain. Not much of these new strains in yellow. However, when we start to look at these, the intervention rooms, we start to see a larger proportion of strains showing up that are new strains to a sink drain. So in phase 2, we start to see a few new strains, but then particularly in phase 3, where we are collecting more isolates again because we're not enacting the intervention, but these isolates seem to be new strains rather than recurring ones. And you see this pattern also repeated in the 5 time per week intervention rooms as well, but it's a little harder to see since there are several weeks where no Pseudomonas aeruginosa isolates were collected, so you can't determine if they would be new or recurring. And so sort of just our main takeaways from all of this work is that without intervention in a non-outbreak setting, these AROs can maintain reservoirs for at least 3 years and cause patient infections. And we were able to observe that this intervention did effectively reduce ARO burden in sink drains. We also were able to use whole genome sequencing to identify that these P. aeruginosa strains may have multiple mechanisms of colonization because we are seeing sometimes recurring strains, sometimes new strains. But of course, further work is always needed to quantify the effect of this intervention on actual patient infections. And so I would love to answer any questions, I think, via email since this is a virtual conference. But of course, I just want to acknowledge and thank everybody who is involved in this work. These were two very large projects, took a lot of people's hard work and time. So just thank you to the Dantas Lab and my PI Gautam Dantas, as well as our collaborators, Jenny Kwan and Carrie Ann Burnham, and their labs and hard work as well. So thank you all for listening. Happy to answer any questions you might have.